Animals are found as long ago as the Ediacaran biota, towards the end of the Precambrian, and possibly somewhat earlier. It had long been doubted whether these life forms included animals. But the discovery of the animal lipid cholesterol in fossils of Dickinsonia establishes their nature. Animals are thought to have originated under low oxygen conditions, suggesting that they were capable of living entirely by anaerobic respiration. But as they became specialized for aerobic metabolism, they became fully dependent on oxygen in their environments. Many animal phyla first appear in the fossil record during the Cambrian explosion, starting about 539 million years ago, in beds such as the Burgess Shale. Extant phyla in these rocks include molluscs, brachiopods, onychophorans, tardigrades, arthropods, echinoderms and hemichordates, along with numerous now extinct forms such as the predatory Anomalocaris. The apparent suddenness of the event may however be an artifact of the fossil record, rather than showing that all these animals appeared simultaneously. That view is supported by the discovery of Auroralumina attenboroughii, the earliest known Ediacaran crown group Cnidarian, 557 to 562 Maya, some 20 million years before the Cambrian explosion, from Charnwood Forest, England. It is thought to be one of the earliest predators, catching small prey with its nematocysts as modern cnidarians do. Some paleontologists have suggested that animals appeared much earlier than the Cambrian explosion, possibly as early as 1 billion years ago. Early fossils that might represent animals appear, for example, in the 665 million-year-old rocks of the Trezona Formation of South Australia. These fossils are interpreted as most probably being early sponges. Trace fossils such as tracks and burrows found in the Tonian period, from 1 GYA, may indicate the presence of triploblastic worm-like animals, roughly as large, about 5 mm wide, and complex as earthworms. However, similar tracks are produced today by the giant single-celled protist Gromia spherica. So the Tonian trace fossils may not indicate early animal evolution. Around the same time, the layered mats of microorganisms called stromatolites decreased in diversity, perhaps due to grazing by newly evolved animals. Objects such as sediment-filled tubes that resemble trace fossils of the burrows of worm-like animals have been found in 1.2 GYA rocks in North America, in 1.5 GYA rocks in Australia and North America, and in 1.7 GYA rocks in Australia. Their interpretation as having an animal origin is disputed, as they might be water escape or other structures.